So this COG database was developed by NCBI, and the one we are looking at was this one with 200K proteins, where we started with about half the data set. There are lots of other data which, if we get this one right, we can extend it. And this tells you about these various clusters that we notate, which we show on the pictures, and how actually some of them are closely related, these clusters, and those closely related clusters are in fact interestingly near each other in the protein universe. So it says the universe is useful. It does take nearby clusters and put them in the same place. That's shown here. There's these nearby clusters. These are a very small subset of the protein universe. If we go back here, that's this set. We're looking at this region here in detail. And you can see these clusters are all actually clustered and pretty near each other. Here we have blue, green, red, purple, and these are the ones which actually are functionally similar. And so the fact that they occur together in the protein universe is nice. Note that some of these clusters, like this yellowish one, it is really not so so clustered. It has there is some clumping, but there's also some spread. What that's due to is not obvious, and maybe the data is just not correct. Here is a comparison of our clustering technology with other clustering methods, and we have the blue one here is the spectrum of the clusters, which we got from our technology. And here we have various other methods, so-called CD hit and uclust. These tend to work by seeding and then growing the seeds and then finding the clusters you get by growing the seeds. And they tend to give you too many clusters. The deterministic annealing starts with one cluster and gradually refines it until you get, in this case here, for the metagenomics sample, 188 clusters. Now that's the end of that particular discussion. Let's just take a few remarks about generative topographic mapping. The huge advantage of that mapping is it's not of order n squared. It goes like the number of clusters times the number of points, whereas MDS goes like the number of points squared. So as long as the number of clusters is much smaller than the number of points, it's generative topographic mapping is better than MDS, is much faster, and it tends to give you better answers than the other well-known method for vectors, which is principal component analysis, which divides the space into its eigenvectors. This is a 12-dimensional space with 1,000 points, and in fact, this data is provided with the plot.viz demonstration. Here's some example from chemical informatics where we're taking 1 million chemical compounds in high-dimensional space using GTM to map them to 3 dimensions. And again, this particular example is also in the plot.viz demo, and it shows you even the visualization rotating. So now we come to a totally different topic, one that's actually related to what we discussed in the search, but it just illustrates because it uses generative topographic mapping. And what it does is it takes all the documents in an AP data set and chops them up, and it associates them with topics. Then if you associate things to topics, then you are effectively mapping all those documents into a vector space, and then you can take that vector space and map that vector space into 3 dimensions, and then you get a plot of this like this, which shows striking clustering. And here's actually you get even nicer answers if you have 20 topics and you map those 20 topics also into 3 dimensions. Here we come back. Here, this is just a spectrum of clustering results, which we've actually commented on already. If we look at clustering, we We have various types of clustering and various types of data structures. Here we have some medical data in 4 dimensions. When we project it into 3 dimensions, we show clear structure. Here we have pathology data in 54 dimensions. When we plot it into 3 dimensions, we find regions, which are these colored regions which are found from the clustering, but there's no striking separation. And finally, at the bottom here, we have some LCMS mass spectrometry data. This data has got dramatic clusters. This is actually intrinsically 2-dimensional data, and we see clear clustering structure. And so this just ends up by pointing out that the visualization helps to tell you what you're doing. If you didn't do the visualization, you wouldn't really know whether there were clusters there, because it's just too hard to really understand without visualization. This just tells you what you're doing, and I believe information visualization is very important. Here's some other examples of multidimensional scaling to do so-called phylogenetic trees. Here's a couple of hundred sequences, and we use those sequences to form a phylogenetic tree, which is a way of forming a tree structure, and then we map all of that to 3 dimensions and show the tree in 3 dimensions. And here's a more complicated example with 2,000 sequences, and I should say that this is slightly speculative, but it illustrates the power of visualization. And although I don't show it, it shows you that some approaches just don't work very well. The visualization makes it very clear that the tree doesn't seem to represent the data very well. So this is just a way of checking what you're doing makes sense. So that's the end of my discussion of health informatics. We went from some really cosmic issues about making the health data of the world more transparent so people can make informed decisions. We discussed linking all possible forms of data from insurance companies to clinical medical records to genomics to social networking data to drug trials, and putting it all together so that we will be able to make good, healthy decisions, and we can all live longer and enjoy more MOOCs and educate ourselves. Thank you very much.